Welcome. Well, as you can see, it's been a few months since our last video recording. I've gained a little hair on my face and I've lost a little hair on my head. And we're here today to take a look at the Stevenson's Research and Technology Center at the University of Oklahoma. Now, this facility was designed to bring together scientists that do somewhat similar research, but also take a different angle to it. So there's a potential for them to collaborate and maybe even create a whole new science. Now, this has worked really well for us. We have the laboratories of molecular anthropology and microbiome research here in this facility. And this is a place where we study ancient DNA. And here to talk a little bit about ancient DNA is the co-director of our laboratories, Dr. Christina Warner. Hi, Christina.
Thanks, Cecil. It's such a pleasure to be here today, and I'm really excited about taking you on a tour. But before we get started, I want to just say a few brief introductory words about ancient DNA and what it is. Sometimes we have evolutionary questions that it's very difficult to answer with only the living descendants of the time period from which we have these particular questions. This is where ancient DNA becomes really useful. Ancient DNA means going into the archaeological or paleontological record, finding traces of remains and extracting DNA from them. And we can read the sequences of those DNAs and investigate these evolutionary questions directly. But working with ancient DNA has its difficulties. As you might imagine, being so old, this DNA is degraded and very sensitive to contamination from the modern world, which is all around us. And so we have to build a special ancient DNA laboratory that is built to keep modern DNA out and to protect the ancient DNA inside. So I'm going to take you on a tour so you can see these highly specialized facilities that we're lucky to have here at the University of Oklahoma.
So come on. Let's do it. Oh, good. The lights are on. I can't wait to show them this facility. This is the facility from the outside, but we're going to see it from the inside.
Now, this is a brand new facility. As we go through, you'll see there's no equipment inside, and that's because it's just been built. We just had a major upgrade, new facilities, new benchtops. We're so excited.
Well, one great thing about our open office area is that we can actually see the ancient DNA facility from our offices.
And here we have two of our graduate students who have been conducting research in the ancient DNA lab, Ali and Andrew.
How's it going, guys?
Good. They're currently analyzing their data from different projects that they've been conducting, but let's go ahead and take a look at the inside. Welcome to our new ancient DNA laboratories. I'm so excited to give you a sneak peek of what is to come. Now, normally in this laboratory, we require specialized protective clothing, not to protect us from the materials, but to protect our ancient precious materials from ourselves, from the DNA on our own bodies. But since this laboratory is still under construction, we can go ahead and come through in our street clothes because it hasn't been fully sterilized yet. This first room that we're in is our storage room. This is where we're going to be keeping all of our supplies, our chemicals, and some of our samples as they come in the door from our collaborators from around the world. The entire laboratory, all put together, is 6 rooms. And as we proceed through each room, it becomes cleaner and cleaner and cleaner so that we can finally, in the last 2 rooms, those are the rooms in which we analyze our ancient DNA. We actually pull the DNA out of fossilized bones and other materials in order to analyze the DNA sequences. Those rooms have to be extremely clean, and they're far in the interior. But let's go ahead and take a tour through each of the rooms in the order in which a sample is processed. So as I said before, this is the first stop of an ancient DNA sample in our lab, where it gets stored and cataloged. Next, we moved into this room, which will be our photography room. Here, we'll photograph the samples and maybe even subsample them into smaller pieces that are more manageable to analyze. You'll also see that here behind the door, we have a changing room, and this is where analysts come in and change into their specialized clothing that they'll be wearing for the rest of the lab. Now, after this laboratory, we move into our microscopy room. Sometimes we need to, before we conduct our analysis, look at our samples a little bit more closely. And to do this, we'll have the aid of microscopes to help us select exactly which parts of a sample we might want to analyze. Next, we proceed into the wet lab. This lab has a sink, and it also has a specialized chemical fume hood and biological safety cabinet that's necessary in case some of the analyses that we require involve chemicals that are volatile or dangerous. This is a safety precaution here that protects us from some of these chemicals that are necessary to extract DNA or to manipulate proteins, which we also do in this lab. Now, standing here right in front of me are our 2 cleanest rooms in the whole laboratory. This is really the heart of the ancient DNA laboratory. They're identical rooms, and on the left side, we'll be processing ancient microbiome samples. So samples from dental plaque, dental calculus, and also coprolites, which is fossilized human feces. And on the right side is where we're going to be processing ancient samples from dentin and bone. This is where we get material to understand the host genome, the human genome. And we'll do that in this room right here. We have special laminar flow hoods that protect the sample to an even greater degree. This is a very, very clean environment. And we also have chemical fume hoods that are mobile on wheels, again, in case we have to use any dangerous chemicals during this process. So after we extract the ancient DNA molecules, we need to prepare them for sequencing. We do the first step of that here, but the next step happens in the modern laboratories. So let's go talk to Dr. Lewis, who will explain how that works.
So the samples from the ancient DNA facility leave in a protective chamber, sealed so they can't be contaminated by modern DNA. Then we do some chemistry on these samples. The ancient samples are amplified by a technique called the polymerase chain reaction. After the polymerase chain reaction, the ancient DNA template is now modern DNA, modern copies of DNA that match that ancient template. Those modern samples, those modern materials can be DNA sequenced and analyzed in all the different ways genomics and genetics has available to us to study in science today. So thank you for joining us on this tour of the ancient DNA facilities within the laboratories of molecular anthropology and microbiome research at the Stevenson's Research and Technology Center at the University of Oklahoma.